Welcome, everybody. Thank you all for joining us for this very special episode of the Stem Cell Podcast, Pathway to the Clinic with BlueRock Therapeutics. I'm Dr. Daylon James. This is Dr. Arun Sharma, and we are the hosts of the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. We're here at BlueRock headquarters in Alexandria Center for Life Science tonight, where I saw a seaplane take off in the East River. I lived in New York my whole life. I knew they were in the air, but I didn't know they landed right there, which is pretty amazing. So I'm already good for the night, but hopefully we can satisfy you guys as well. As I said, we're in front of a live audience here in New York to discuss bemdaneprocel. Oh my God, I screwed it up right out of the gate. Bemdaneprocel, sue me. A stem cell-derived investigational cell therapy designed to replace the dopamine-producing neurons that are lost in Parkinson's disease. The results from a phase 1 clinical trial were released back in August, and I think we were all, the world, was very impressed, not only with the safety profile, but some really encouraging early results about efficacy. And really, for me, just the culmination of all these years of promise finally coming to fruition. So it was very gratifying. We're going to talk about that. We have the team here, some of the team involved with the work here to chat about it. And without further ado, I'm going to introduce our guest now, Dr. Stefan Irion, who's the Chief Scientific Officer at BlueRock Therapeutics. Stefan leads the research organization at BlueRock Therapeutics since joining BlueRock in 2017. Stefan has taken on a number of roles in support of BlueRock's scientific goals, developing CNS strategy, advancing the DA01 program through the IND clearance, advancing multiple neurology pipeline candidates to the next stage of development, to building and expanding a cross-site and cross-functional research team. Prior to BlueRock, Stefan was part of a leadership team at Memorial Sloan Kettering that developed MSK-DA01, the current lead program at BlueRock. Stefan worked as a senior scientist at Hyperion, developing iPSCs prior to joining MSK. Stefan received his MD from the University of Tübingen and did postdoctoral fellowship work at both Mount Sinai School of Medicine in New York and University Health Network in Toronto. Welcome to the show, Dr. Stefan Irion, please. And of course, we also have Dr. Viviane Tabar, Chair of the Department of Neurosurgery at Memorial Sloan Kettering Cancer Center. Dr. Tabar is a founding investigator at BlueRock Therapeutics, a neurosurgeon and professor at Memorial Sloan Kettering Cancer Center in New York, and holds a joint appointment at Weill Cornell Medical College in the Departments of Neurosurgery and Neuroscience. Dr. Tabar serves as a co-principal investigator of a large consortium grant aiming at the development of a clinical trial for grafting human embryonic stem cell-derived dopamine neurons in Parkinson's patients, and has focused major efforts on development of human ES-derived oligodendrocytes for brain radiation damage. She's also one of the 2 neurosurgeons that participated in the phase 1 trial of bemdaneprocel. The worst. She received clinical training at the University of Massachusetts. I'm not even drinking, by the way. Everybody else on this panel is drinking. Not right. It's not fair. She received clinical training at the University of Massachusetts and at Memorial Sloan Kettering, and research training at the National Institute of Neurological Disorders and Stroke at the NIH. Now, to be clear, Dr. Tabar is speaking with us here in her role as a physician scientist who has guided the work. And while she does have a financial interest with BlueRock, she is not speaking on behalf of BlueRock and is not really going to be speaking about BlueRock pipeline at all. She's here to talk about her work and her role in this pivotal step forward. Welcome, please, Dr. Viviane Tabar.
All right, so let's dive right into it. We'll start off with an easy question for you, Stefan. Everybody here in this audience knows what BlueRock is all about, but for our listeners at home or in cell culture hoods around the world, could you describe in your own words what BlueRock Therapeutics is, how it came to be, and maybe what sets it apart in the stem cell field?
Yeah, of course. So in a nutshell, we are a regenerative cell therapy company that harnesses the power of pluripotent stem cells, which this audience and the Stem Cell Podcast audience is well aware of, to bring new therapies and new hopes to patients that are suffering intreatable, intractable diseases today. We were founded late 2016. It was really we refer to as a moonshot effort. We raised one of the largest Series A funding rounds invested by Leaps, the venture arm of Bayer Pharmaceuticals, and Versant Ventures. Raised $250 million, which is a lot of money, and at the time was even more money, to start this moonshot approach, to really build a platform company around pluripotent stem cells. So not just saying, I can treat diabetes, and you build a company around that, but really thinking, what are how can we best leverage this potential and build a whole company around it? So we make this investment in understanding and how we grow and expand pluripotent stem cells, how we might genetically engineer them to then have multiple therapies that we can use to treat patients. So the founding programs that came in was a program by Gordon Keller and Mike LaFlamme up in Toronto, how to make beating cardiomyocytes for patients that have suffered heart attacks. I see you high-fiving that, Arun, yeah. And obviously the program that I was involved here with Viviane and Lorenz and Mark Tomishima, who's in the audience, to bring dopaminergic neurons to patients that suffer from Parkinson's disease. So that was the early days. We started the programs here in New York and Toronto and later Cambridge. And then about 2-3 years later, we were acquired by Bayer Pharmaceuticals, all right. And today we're operating as a wholly owned, independently operating subsidiary of Bayer Pharmaceuticals.
So yeah, you mentioned it's Parkinson's disease. I mean, that's what we're talking about tonight in terms of the drug and the innovation here. But the disease is what this whole conversation is about. That's the impetus, starting with you, Dr. Tabar. I mean, notwithstanding Lorenz's grace on poop duty with the bull, what brought you to your study? This, I mean, monumental lift, the ambition it took to go after a disease of such scope and with such a challenge. Why'd you jump on board with that ambitious challenge?
Yeah, good question. I sometimes wonder, except in this last year, I stopped wondering because we're just so incredibly excited that it came to be and that it was successful, even though we're still in early stages. Look, I loved the brain from the beginning. I became a neurosurgeon because back then, no offense to any neurologists in the audience, I felt that we didn't have too much to offer to patients with devastating disease. So I figured, okay, maybe we can remove some tumors, take out some blood, some, brain trauma. So that's what drove me in sort of my professional career. But in general, in science, it's really, honestly, all about vision and perseverance. And that's what I learned in the last 20 years. I went to NIH, to Ron Mackay's lab, because I heard him talk about stem cells in the brain, which was not what our medical school books taught us. And I just got, in my medical school application, I started with this sentence that I never forgot, which is, once your brain is stretched by a new idea, it never regains its original dimensions. And that really describes the trajectory. So in the beginning, we were looking at rats, at trying to scale up fetal progenitors. And that was, the first experiment with Lawrence and me. And then we just happened to be a really good team where I was already, even when watching rats, I was already thinking how that would play out in a person's brain. And it sounded truly wild, really. And he, of course, came to it with a true scientist's mind, but also with, what otherwise you would call stubbornness, but what do we like to call it, perseverance, of that thing, making it happen. And then it was a series of good decisions. One of them was hiring Stefan as our project manager. And the other really was finding ourselves in a cancer hospital that was willing to let us explore the science. And I can't say enough for all of you who mentor young people, who direct labs, anything, your homes, that you should always listen to exciting ideas and see if there isn't something there that you can push through. Luck favors the prepared mind. So there's a mix of being prepared and thoughtful and being at the right place, right time, right people.
You mentioned Stefan. Stefan, to you, I mean, you were at Irion working on iPSCs, not far from this, but not exactly right in that wheelhouse. What brought you to Parkinson's? And also, as a follow-up for maybe both of you, what do you think were the most instrumental steps that were critical to reaching this point? Just one. I mean, we're going to talk about so much in this conversation, but if you could think of one critical watershed.
Yeah, for me, it was, I had fallen in love with the stem cell biology through my postdoctoral training altogether. And so my first industry job was with this company, Irion. Didn't carry my last name in it, but close. We were trying to really, for the first time, leverage iPS cells that just sort of became something to use them for drug discovery. That was quite exciting. And for a variety of reasons, moved to New York, and I knew that Lorenz was there, and I knew him through the stem cell field as one of the leading scientists and smartest guys I knew. I approached him and said, I'm in New York. I love the science. I want to do something here, and I was really trying to look. Can we find something that is not an academic faculty job, but something different? And he said, well, as luck may have it, I just applied for this grant, and it looks like we're going to get it to develop a cell therapy for Parkinson's disease. And he may have not finished the sentence. I said, I'll take that job. And I remember Viviane was very concerned that it may be boring in the beginning. I think for me, it was the vision. I said, there's absolutely fantastic science behind it that I believe, and there's a huge unmet clinical need. And there's precedence in the literature that a cell therapy for Parkinson's should work. And I put all these pieces together, and I said, that's what I want to do.
It's an incredible journey for the both of you, for all of us, really. It's a little bit of luck, but I think mostly hard work. And I think we're all so fortunate to be in the midst of this stem cell revolution. I think we can accurately call it that. That's enabling all of these discoveries and these translational advances. So, before we actually dive into the trial results, I'd like both of you to reflect on the quote need for a biotech company like BlueRock in facilitating a complex trial like this. I mean, in contrast, there are clinical trials that are start to finish conducted entirely at academic centers, right? So maybe it's, a bit of a naive question, but what was it about this therapy, this approach that necessitated BlueRock to be involved in this trial as such a critical partner? It might be obvious to everybody here in this audience, but for our listeners at home who don't necessarily think about cell therapy day and night, like you all do, why do you need a company like BlueRock for this clinical trial to happen?
As you said, you can run a phase 1 clinical trial in an academic center. I think what sets us apart here in a biotech company is really this mission to develop this therapy for patients in need, for everybody that needs to take it, right? So from the very get-go, we're focused not just on, we believe already in the phase 1 trial, but it's really about the next thing, right? So we're investing in scaling up manufacturing. How do we take this to all the patients in need, not just the 12 patients that we enrolled in the phase 1 trial, right? You can throw money at things and make them go faster and in parallel. And it's incredibly difficult in academia to do that, right? When we at Sloan were thinking about how do we take this to the next level, we're thinking, well, maybe there's an NIH grant, but we're talking, and this may sound ridiculous, but small $1 million things, while at a biotech, we were able to really take a lot more to make things go faster. And I think one of the examples is our enrollment was completed within a year, which most people would think is really difficult in a Viviane can speak to this, a complicated surgery, right? That takes some time and preparation. So to get the 12 patients within a year was the effort of not just being in one site, but really pushing to go to another surgical site, opening up the trial in Canada, and all these things. I think that was what we brought to the development of bemdaneprocel, is, by the way, how we say it.
Got that. All right. Let's talk about the results a little bit. I mentioned them briefly, but you guys can elaborate. The bottom line being that it wasn't just the safety, which is the primary endpoint, but there were some efficacy results. And as I said, you guys could elaborate on it. But I want to try and get both your perspectives, starting with you, Dr. Tabar, the neurosurgeon perspective, the patient being the priority. What does this success enable for that patient, both like on a macro level in terms of their life, but also just logistically moving on from this trial and the success? What does it enable you to do? And the next question to ask in terms of efficacy and scale or whatever that may be.
So I have to add to what Stefan just said, that a lot of very good ideas go to rest in the valley of death before they reach the clinic. So founding and finding BlueRock was absolutely critical to this idea becoming reality. I remember, I don't know how many years ago, I wrote a little something in the literature that says stem cells hype or hope. And so stem cells went through this long period of wondering, are they a smart cell? You just put them somewhere and they know what to do, versus are they a cell that needs a lot of help, a lot of direction, and then putting it in the right place. And as we moved on with the science, it just starts dawning on you that, hello, there's going to be a patient here, you're going to put this in their brains, and you've got to do it right. So everything matters. Every step of the way matters. You needed an industry sponsor to support the trials, which the first time I looked at what the budget might look like, it was stunning. This is an intervention that requires us to do surgery on both sides of the brain, pass a cannula 3 times on each side, put in a total of 18 cell deposits. We, went really overboard with emphasizing the safety. We did this in an MRI-equipped operating room. We didn't leave anything to chance. One of my partners in crime, in addition to Lawrence, is Kenny Yu, who's sitting back there, who was this, young, smart neurosurgeon who was sort of willing and all gung-ho planning the trajectories, while I was worried about if the cells are going to be okay, with Mark Tomishima bringing them back and forth from the facility with his little igloo. There's a lot of detail, and you can fail on the detail. I mean, these cells that Lawrence made could be fantastic, but if we don't get them in the patient's brain at the right place in good condition, they wouldn't work, and the trial could fail. So it was all about attention to detail and about a really genuinely great team who really believed that this could happen. So that's for one. From the patient's perspective, I will remind this audience that we got FDA clearance maybe, 10 days before the EUA for COVID vaccine came. I'm not making this up, but I was putting a turkey in the oven when I looked at my phone, and there was an email from the IND office at MSK saying the FDA has a question, can you respond? When does that happen? Over Thanksgiving. And the person who was telling me this has been doing this for 20 years, has never seen that. The FDA goes home for a long time around Thanksgiving. So there was an emergency in the country. There was a pandemic, and here we are wanting to cure Parkinson's at the same time. So we got this clearance, and then it was like, what do we do now? We got the clearance, but we're in New York City, the second center of the pandemic. The most impressive thing about all this to me was the enthusiasm of the patients, who all came in equipped with a preprint from STEMCELL that had our preclinical data, and with links to webinars that Lawrence had done, or I had done, or somebody else had done, or Stefan, about this. And they were all in. BlueRock supported them to be in the city for 28 days during the pandemic to have the intervention. And they were truly inspiring. I have a whole shelf of things they sent me, angels, T-shirts with their trial numbers, etc. And, those patients knew what they were getting into, and they looked me in the eye and said, yeah, I'm willing to do this, even if I don't benefit. This is so exciting. We've been waiting for it. It was genuinely a, kind of feed-forward, positive feedback loop to get this done. And, yeah, we did 1 patient a month, which is the maximum we could have done by our protocol. in the middle of a pandemic. And that took, I know it's cliche to say it took a village, it took a town, a city of people to actually make it happen. But most importantly, it took the patient's trust in the science at a time when we were challenging the science of the vaccines, right?
Well, you said the patients were inspiring. I think you inspired the patients. I think that's the therapy is really, I mean, the hope and the promise is just outsized. Stefan, to you, I mean, for BlueRock, a lot of attention. The world's looking at this therapy. This is like the first big scale, not the first, but the first of this scale. What does it mean moving on from here for BlueRock? Can you just talk about that a bit?
Yeah, I mean, it is absolutely critical for us and the whole community that these trials of truly innovative cell therapies succeed, right? So we're looking equally around us where others are in this moment in other therapies, but also in the Parkinson's field. For us in particular, of course, this was one of the founding promises, one of the moonshots that we wanted to take is to restore and re-innovate the brain that has suffered from Parkinson's disease. And I think what we're seeing today in our early trials is exactly that. It's really tantalizing. We see engraftment, and we talk about this later, of the cells. And this is what we came for. So this proof of concept, this human proof of concept, is so critical for our success because it's going to help us rally the community behind other cell therapies that have a very similar principle. And it allows us, our funder Bayer, to believe in what we do and continue funding this work, to bring it not just to Parkinson's, but other therapies, but importantly also, like we're planning to do, to move this Parkinson's trial beyond a clinical trial to an actual therapy.
So actually, let's talk a little bit more about exactly that. Shifting away from Parkinson's for just a little bit, you mentioned some of those other therapies that are, in development potentially. These are mostly preclinical studies at the moment, but, they're for other neurodegenerative diseases and other options, other cell options in terms of cell types, such as microglia, oligodendrocytes. But you also have preclinical studies, and I was just looking at the website for BlueRock. So there's preclinical studies going on in ophthalmology, immunology, and in my favorite field, cardiology as well, right? And I know you can't say a lot about these preclinical studies, but could you talk just about these other areas of interest that BlueRock has and what excites you in terms of these therapeutic approaches?
I think the next big thing for us is our program in ophthalmology that we have in a collaboration with Opsis Therapeutics. There we're trying to make photoreceptors and restore vision in patients that have lost it due to genetic causes of blindness. And for me, that is fascinating. If we can bring even light-dark reception back to patients that are blind, that's fantastic. I personally work and run with people with blindness, and just to help them is personally very motivating for me. So that's exciting. That science speaks to me. It's very similar to what we see in Parkinson's, where we can bring new photoreceptors to those that have died. And this is particularly interesting because it goes beyond a certain genetic mutation. It has the potential to treat all genetic causes of blindness, or at least many of them. So that's exciting. The next one for me here internally at BlueRock, our second program, the original moonshot, was this idea to remuscularize the heart. A completely different challenge. We have to make, billions of cells to restore heart function, right? To engraft them, they have to couple electromechanically so they don't cause arrhythmias. There's a completely different challenge that we've seen than we have seen with Parkinson's. But the fundamental principle to bring cells back and treat the disease that way is the same, right? In all other therapies rely on treating what's left and making that work better or differently. Here we're actually bringing fresh new cells back. So that's part of our pipeline on the cardiac side. And then our earlier research programs around microglia, absolutely fascinating cell. Can riddle off numerous indications where microglia play a role, neurodegeneration, some rare genetic causes. We've talked about some preclinical work we've done in lysosomal storage disorders, where typically people use bone marrow transplantation to bring the enzymatic function back, and we're able to show that in these preclinical models we can do this with microglia, really overcoming one of the hurdles to bring this enzyme into the brain very early on, before neurons die in patients. These are some of the programs we're pushing forward, and the team here in New York is mostly of our neuroscience research is working on. Pretty exciting times.
Yeah, I mean, the most exciting thing to me, I think uniquely in science, where there's so many haters, I feel like everyone's rooting for you. I mean, everybody wants this to succeed. And Dr. Tabar, you said it, valley of death, you needed this to be a success. Stefan, you said it, it was really critical to inspire confidence, both from funding agencies, but also everyone else, right? Dr. Tabar, so given that, and we alluded also to there's a lot of other trials going on, right? And I think everybody's rooting for them to succeed too. We're all on the same team, right? On some level. But with that in mind, and with this success being kind of like, I don't want to say unleashing everybody, but it's real. And I think everyone can use it to convince whoever is the gatekeeper that they should be moving forward with whatever the endeavor is. So with that in mind, do you think that these next 10 years is going to be like a bonanza or a true breakthrough? Or do you think that with your experience and all the rigor that it takes, that like we should temper our expectations and revise our expectations to be a little bit more of a long-term kind of drip out of these big news stories?
Well, it's a mix of both. And so the success of this trial, and by that I mean the trial meeting its endpoints of safety and giving signals of efficacy, which is all you can ask of it right now at 1 year. We are already well ahead of what has been tried before with fetal tissue, for example, in Parkinson's disease. So that's truly, truly exciting. And like you said, both of you earlier, I mean, this is going to be a decade of incredible advances. I am extremely enthusiastic, but also very pragmatic. The brain is a very complex organ. I cannot speak for the heart. I think it's secondary to the mind. But in terms of complexity, sorry, it's equally important. I take that back. But in terms of complexity, if you succeed in the brain and you get a signal, and let's think about it. What is this signal? The signal means you have put in a fetal neuron and it has connected with your own synapses and is now part of you and trying to repair something that wasn't working well. I mean, I wouldn't be too unrealistic if I said from here on the sky is the limit. At the same time, that proof of concept is extremely important for the patient, for the industry, for the physicians, the scientists, and I do believe it will inspire everybody. But we also temper it with maintaining our rigor as scientists, and our expectations should be really driven by the science and not by anything else. For example, we often talk about making truly fetal dopamine neurons in the dish and replacing them in a patient who is 70 or 75 years old. And, we have to give that therapy time to function. And we have to, continue our lifelong learning. And I'm very excited that there will be other trials, other cells, other groups, new ideas that will only serve to make the therapy better. One of the hardest things, I think, for our team, when we were still, all of us, Stefan, Mark, Lawrence, myself, and many others, only in the academic space, we were already sort of thinking, we had a hard time locking down our product and stop thinking about making the protocol better and the differentiation better. And that's when you realize that you made that transition to go to clinic, and we kind of went into it kicking and screaming. But we already are thinking about how we can even be better. So, you always need the pragmatism of the clinician, the excitement of the scientist, and of the patient.
Yeah, it truly is a team effort, and we can talk more about that heart versus brain debate offline. My department chair is a neuroscientist, so I'm not new to that debate. To be fair, the heart, it's just a pump, okay? It's just a 4-chamber pump. Fine.
A very good one.
It's a decent one. It's decent. It only pumps like 3 billion times or something like that, right? So I actually wanted to take a step back, and you highlighted this briefly already, but I just wanted to do this again because I think this is so important. The most important members of this clinical trial team. Can anybody guess who they are? It's the patients. The patients are the most important members of the trial team. I mean, these are brave individuals affected by such a devastating disease and who underwent brain surgery, right, to actually receive this investigational stem cell therapy. And I mean, Dr. Tabar, this is what you do professionally every single day. But for those of us like myself who don't do brain surgery every day, this procedure is daunting. It's scary to think about. Can you reflect again on just the courage of these individuals and their families as well, receiving this therapy? Because this is ultimately about them. You mentioned they had copies of stem cell coming that they're bringing with them to their therapies, but I just want to emphasize the importance of these individuals who are the most critical member of the trial team.
Yeah, that's really, you're right. In all of this, those are the most important elements of all that. We often think of a physician as sort of a bit of an authority figure, someone we kind of go to ask for advice, help for something very, very precious like our health. But actually, it goes both ways. It's a reciprocal relationship. For the patient to come fully trusting, to say, yes, you can do this. You can drill a hole in my head on both sides and pass your needle a few times each. And some of them wanted me to count how many times we would do that and to put in those cells. It fills you with awe and a sense of responsibility that, all of a sudden I felt that I was a little bit, like 2 steps away from the science team who really wanted to know if the cells were alive and if the mix was good and the temperature was good and all that was important. I'm like, oh my goodness, here I have this patient with their head open and I have these cells in there. Better be something good for these patients. And then you had to go tell their families how it all went. And I will be honest in saying that the first few were, like everything, took longer than we expected, and because everybody wanted to do the best job they could. But really, those patients were very, very honestly, very, very supportive and inspiring and grateful. And I was quite moved. I mean, I deal with patients all the time, but these are patients who have been following the science and look forward to the future and who really never underestimate patients. They really know more than you think. They know that they are participating in an experiment, a grand experiment, but an experiment nonetheless. And they go into this fully aware and very excited to be part of it. And many of them confirmed that even if it does not work for them, because this was a trial that had 2 doses, and sometimes I had to answer questions about, well, which one is better? Should I wait until you get into the higher dose? Like, how do you answer that one? I'm not supposed to say anything. I don't know. Also, how would I say? There's a lot that goes into it. People ask intelligent questions and still give you their trust to do what's right. And I think I'm a physician, I'm used to this, but I can really say that the entire team, the lab members, the people who were bringing the cells to the operating room, BlueRock, everybody was on board to do what's right for the patients.
Right. I mean, they're super courageous in a very vulnerable position, but give yourselves and the team a lot of credit too. I mean, it takes a lot of courage, I think, because they look up to you and they believe, I think. You're a brain surgeon, for God's sakes. They believe that you know what you're doing. If not in life, at least in the operating room.
Yes, most of the time.
So they believe, you can tell them anything. They're like, okay, doc. But you guys are in space, right? And like, let's talk about it. You had to kind of develop de novo techniques. This is stuff that's never been done. I've listened to your talk at NISA, Farun listened to your talk in San Diego a week ago. Like, the details are really critical because I think they underscore how anything new requires a little bit, I don't want to call it like a DIY approach, but you had to create new tools and you had to, can't try it out, right? This is the first patient has to be as good as you can possibly do, as you were alluding to. Can you elaborate on some of those? I think it's really interesting for the scientists and the physicians probably and surgeons, like what kind of, for example, type of like novel methods, like the minute volumes, etc. Could you elaborate on some of these, the idiosyncrasies of the technical procedure that are more daunting, I think, than people realize?
Yeah, well, the first one was, holy cow, Mark, is this one sterile? The cold, so Mark had to bring the cells from the GMP facility where they were thawed. And they come in this box, insulated box. And then, I have to stop for a moment just to give a shout out for the nurses who helped us make this happen. I couldn't have done it without them. Nurses, OR technicians, they were equally excited. But, you have to comply with very, very, very specific rules. You in the lab, you might assume that the cold block or the ice bath, etc., are okay to use, but not in the operating room. So you had to think out everything. I mean, we trialed quite a few things right here in BlueRock headquarters. How do we take out the cells? How do we suspend them? What has to be sterile? And oh my gosh, you do need pipettes. The OR has no pipette. All these little details. You couldn't really do it if you could not comply with very strict rules. And then when it came to injecting these cells, which, worked great in the rat to give a few microliters, but now you got to give a few microliters in a much larger brain. Well, neurosurgery, delicate and precise as it may be, has not developed instruments that can deliver 2 or 3 or 1 microliter of cells. There was a leap of faith to be able. So we knew some of that ahead of time. We modified existing cannulas to diminish the dead space because we're delivering very small amounts. And then, I'll be honest in saying it was reassuring that BlueRock said, oh yeah, you can use up more cells. It's okay. And that's a lot of money, more cells. But you had to do it right. We were so conscious that prior trials have failed, and even they could have failed on the detail. So yeah, the cannula, I like to show this picture of Kenny checking if there's a drop of cells coming out of the cannula, because there's such small amount. You could be injecting an air bubble for all you know in the patient's head, for example. And then your trial would fail, not because the cells aren't great, but because the technique was suboptimal. So we went, all bells and whistles. We used MRIs. We planned our trajectories as if our life depended on them to optimize safety so we can see a signal if there was a signal, and thankfully there was.
Yeah, it's just seeing those results and seeing them presented in San Diego last week. That was the first time I had seen them. It was just, again, I'm in cardiovascular research. I don't do neuroscience, but even to me, it was just so astounding, and so just as just somebody in the stem cell field, I was just so appreciative of what I was able to see. So thank you, and thank you to the team for all this incredible work.
The heart is next.
Heart's next.
You know that.
Yeah, fingers crossed. Absolutely. So, shifting gears a little bit, more kind of, I guess, big picture. It's an interesting time in biomedical research, and especially when it comes to the trainee side of it. I was a trainee not too long ago, so I feel like this is a really important question to ask. It's in academia, a lot of folks are shifting towards biotech. There's great opportunities in biotech, great opportunities at BlueRock, for example. I'm a brand new PI, so it's tough to hire good postdocs a lot of times. A lot of folks are going straight to industry, which is great, which is a good thing. I'm not saying it's a bad thing. But I just wanted to ask a question about career development. What is BlueRock doing to facilitate the development of junior scientists? Because this is a career path that wasn't necessarily available 10 years ago. So, Stefan, what is BlueRock doing in this regard?
Yeah. I think there's something for everybody, right? Like for some people, when I talk to them, I would recommend they stay in academia, right? There's this job here is not for everybody, right? I think what we can offer for people, it's working on a much larger common goal. You wouldn't find yourself as a postdoc running your own project, doing everything soup to nuts, right? So we train people and help people to develop skills, how to work in a mixed environment of different skills and leveraging them really. And I think that's where the power comes in, how you can move things forward, right? By not collaborating with somebody that doesn't want to. You're actually collaborating with somebody that works in the same company and follows the same goal as you have. So here, that's a skill that we teach, right? A lot of people, when they come out of their postdoc or as a student, you don't have any experience how to manage a person, right? How to motivate a person that works for you. So we put an effort into training our next generation of managers and leaders. Internally, we have, maybe cliche, we have two programs. We have a BlueRock Star Leader program and a Leader Plus program, the sequential programs that we enroll people. And we work with outside firms as well to provide additional training. How do you, how to manage people? How do you manage projects? So I think you get an exposure to a broader facet of skills than you get in an academic setting.
So yeah, let's talk about that in terms of the growth, right? Building the company and the young talent you bring in, and how this result impacts the bigger picture. I mean, this, as you said, you've got a whole constellation of targets. What's like your best case scenario in your mind's eye, how this fits in the bemdaneprocel? Did I do it? Nearly. How that fits in, how this impact result, like, where does it fit into your mind's eye? Best case scenario in terms of growth of BlueRock, and not just the company, but about these therapies. Like, what's your best case?
Yeah, this goes all the way to commercialization. That's the best case. That's the vision. For me, this is now no longer a question of like, do we see what we saw in the phase 1 trial? It's really about how do we enable this to become a commercially viable solution. I think that's where we put an enormous amount of effort behind, and that's where you also need to add a lot of resources behind. And that's part of what we have done here at BlueRock, is just prioritize the effort around bemdaneprocel to the full extent. That comes with a lot of resources to go behind that. And I think that's where I see the future for the field as well. You asked earlier about, what does this do to the field? I think it now enables us to think beyond these phase 1 trials. We now really need to think about, how do we make this a therapy? There's so many questions. We've seen it, we're fortunate enough to have some lessons from the CAR T-cell field now. When we think about how to manufacture these at scale, how to bring them to all the patients, how to ship them, how to make them affordable, right? All these questions are now worth being answered because before it was a dream, and now we have the reality of the data set that we have, and it allows us to move forward and think about the next steps.
Great time to be alive. Dr. Tabar, to you now, talk about the patient. Best case for the patient. I mean, I know it's a watershed already, right? Because the fetal grafts, like, it was about a year that you started to see a little bit of dyskinesia. I mean, I'm wrong on that, but I feel like I don't want to say anything out of the woods, but it's encouraging, as we've been saying over and over. So for you, best case for the patient, what are we thinking about? Like, I know full recovery seems like a miracle, but do you think that's in the constellation of possibilities?
Yes, certainly in the constellation of possibilities. I think these are, baby steps, monumental as they may be. They are early steps. I think best case scenario is that the patient will benefit from improvement in their symptoms, particularly their motor symptoms, that their longevity with good quality of life will be longer. But we are already thinking how we can approach this really complex disease that is not only motor, that eventually, after a while, can impact the entire brain, diminish cognition, how we can actually provide an even more comprehensive solution for this disease. But unfortunately and fortunately, you have to go step by step. This takes a long time to mature, and it's important for the field to keep an eye on that, to remain positive, but also be very pragmatic. That these therapies have incredible promise, and here we are on having already one step under our belt, so to say, in the right direction. But there's a lot more we can do, and since we know how to do this, we can do it faster, more targeted.
So I think we're coming close to the time limit here before we get into audience questions. I kind of want to shift gears a little bit because we're talking about the team. I mean, the team is really deserves a lot of the credit for bringing us to this point. And, we always talk about in team building, you need to have a diverse group. And I think you achieve that by pulling together all these scientists from different fields and different levels of expertise. Of course, it's obviously critical, but of course you need to have representation from all different types of people in order to instill that creativity. Saw from the Halloween picture parties that Mark was generous enough to share with me that you have a nice camaraderie here at BlueRock. So the team is critical, and you as a woman scientist and neurosurgeon, 1 in 8 practicing neurosurgeon is a woman. So there's a clear disparity. It's a male-dominated field. And I mean, I don't know that you could speak to broader diversity, but what would you say to a young female neuroscientist looking to get into the field? And or what do you think needs to change in order to balance that, disparity, not just in neurosurgery, but specifically in neurosurgery, but broadly speaking?
Yeah, really good question. So I would say to the woman neuroscientist or neurosurgeon to be, go for it. And be very confident that she will succeed. And I would say for those of you who, run labs, run companies, run groups, that you have to have an open mind. And I get very irritated sometimes when people talk the talk about diversity, but actually don't take action about it. It is not that hard. If you want your team to have people of color, you want Black people in science, you actually know where the high schools are in Harlem, in the Bronx, go and recruit there. I don't take this business about just talking. And I try to practice what I preach. You have to be pragmatic about it. I think nowadays, to be fair, it's rare to encounter very intentional sort of bias. But bias is in our culture. This is quite cliche. I'm proud to say that in the case of our family, that's not the case. But when you ask children to draw a surgeon, often it's a man, not a woman, but my daughter drew a woman. Thank you. It starts very early, and you have to have an open mind, and you have to realize that while all of us strive for excellence, actually you need to make diversity part of that excellence. There are institutions that are brilliant at recruiting the best minds to be their scientists, to be in their company. But if you don't incorporate diversity as an actual surrogate and corollary and goes hand in hand with science, you're actually missing out on talent. And so it has to be incorporated in that culture. And as a leader, you have to take steps to ensure lack of bias. People who come from poorer backgrounds or women who have had to have children, well, their CVs will not look very much like the standard white man CV. Not that there is anything wrong with being a white man. Most of us have been mentored and supported and driven forward by brilliant white men scientists. I would just be explicit. But a woman who had to raise 2-3 children, well, their CV is not going to look the same as the other guy, but she may be incredible. I mean, to be able to multitask, you're already a step ahead. You have to have an open mind. You have to go find talent where you know it is, rather than talk about it. And one important thing as we think about stem cell technologies is diversity among patients. I said that at NICEF yesterday, maybe in a bit of blunt terms, but we don't want this therapy to be therapy for rich people only. It's very hard and costly to make stem cell therapies, but it is not what we're after, to have niche things where you come into the stem cell clinic like you go to some spa. It has to be for everybody. And everybody is not even your neighborhood, your country, it's planet Earth. And that is a huge challenge, actually.
Absolutely. And I think I speak for all of us when I say that having you in a position of visible leadership is so critically important to inspiring that next generation of women neurosurgeons and neuroscientists. So thank you for what you do.
And Stefan, even though you're a white male, you're all right too.
No, tough act to follow. Tough act to follow.
Oh man,
In full support. I think what stood out, and that's what we live by, is diversity is an asset, right? It's an asset, and we have to leverage. It.
Absolutely. All right, so we are nearing the end of our show, but before we let everybody go, we want to open things up to the audience to ask questions. Actually, before I do that, is there anything that we missed that you want us to talk about still before we open it up to everybody else?
We should say for everybody in the audience and that's going to listen to this later in the show, what an amazing time to be in this field, right? I think, thank you both for having this and bringing this out into the community, the stem cell community, and hopefully beyond. Everybody should hear about what these types of therapies can do. It's amazing. I think as a trainee, I'd be ecstatic to listening to these. Well, that sounds silly when I say it, but you know what I mean? Like, this is an exciting time to be a trainee and go in this field and develop these therapies.
Yeah, we say it all the time.
All right, let's open it up. Anybody have some burning questions to ask?
That was amazing. Thank you very much. So you touched on CAR-T and how that's changing the way we approach cancers and things like that. I think there are clearly manufacturing issues though in delivering that to a large population. So thinking about this therapy going from phase 1 to phase 2 to phase 3 to even commercialization, I hope you can get there. I assume there are still massive roadblocks to get to that level of scale. I just wonder if you could touch on that and like what else is going to be needed to enable you to be able to reach that, to deliver this to a large population.
Yeah, I mean, from our side, I like to refer to them as opportunities, not challenges. Makes it a lot easier to digest. But the whole ecosystem around these types of cell therapies is not quite there, right? All the automation that people have developed to make antibodies isn't, thousands of liters of bioreactors. That's not the scale that we're talking about to make millions of cells for transplantation in Parkinson's, right? The producers of the ingredients that make up these cell therapies need to think about how they can lower their costs and make the materials better and cheaper and in larger quantities. That's just to make the cells. And then we need to think about how we distribute these, how do we ensure stability, how do we make it so easy that we don't need a specialized facility to prepare the cells. We don't, we can use multiple surgical centers to deliver the cell therapies and not have to just rely on individual very specialized centers, although we, of course, love working with the centers we work with.
I agree. I think one of the challenges is going to be trying to couple manufacturing and delivery devices and making them, idiot-proof. But that's the key. I'm very optimistic about the field being ready to do that. I think we know the challenges, and I think these are engineering problems that can be solved. We just have to prioritize them ahead of time, because we have to always remember that the success of this therapy has very much to do, as we said earlier, with every step. And only one of them has to fail for the therapy to fail. And that's tragic. Can you imagine? Failing on account of something technical.
Other questions from the audience?
Thank you guys so much. So big picture breakthrough discoveries, like the ones that are happening here, get everyone excited. But I'm curious what advice you have for the trainees and early career surgeries like myself in the weeds of mechanism and detail in our benches and primarily academic class. How do we stay excited and engaged?
Well, you don't love mechanism? Mechanism is awesome.
She loves them too much. I have to say, if you ask me if there's something else to say, well, yes, we're all focused now on the cells and how we got them into the patient, but there would have been no cells if there were no people thinking about the mechanism. So in this audience alone, we have Taewon. Jinghua is not here, but we have some of her team. Those people were absolutely critical to actually making the right cell. We couldn't be where we are today without somebody like you, Kelsey, thinking about mechanism. It's, science is tough. Science is even tougher today, where everything is moving fast and requires your attention, and the attention span is shorter. Science requires you to tough it out for 5, 6, maybe longer years to see if your idea is right or wrong, and challenges you to be open-minded about whether your hypothesis was the right one or whether you proved it to be null or, So we need you to be focusing on mechanism. We each have our strengths and like Stefan says, look at it as an opportunity. This is all based on very rigorous science, and very rigorous science thinks of mechanism and asks why and why not.
Not to mention, I think Stefan or whomever is thinking about the next wave of BlueRock employees. You want people who are classically trained, I assume. I mean, you don't want people just for their thumbs. You need people who are going to think. I don't want to put words in your mouth, but how do you feel about this next generation? Do you think that the classic training paradigm is still preparing scientists to fit well into industry?
Yeah, absolutely. I mean, I think if you think about where some of the greatest discoveries over the last couple of years came from, there were basic scientists, like CRISPR Therapeutics, or CRISPR enzymes some people thought, well, why are you studying this, right? And now we have therapies in the clinic that are making differences in patient life. So I wouldn't overthink what you're studying is not relevant. You never know until you know what the potential it has. For us as to recruit scientists, we like that the basic training is the same, right? Like it's how do I design a good experiment that has parameters that I can control to then make a conclusion and move it forward. You learn that basic principle of how to execute a good experiment in your PhD and in your scientific training. It doesn't matter if you're studying a mechanism and then later apply it to a biotech company or not.
Keep on with the mechanism.
First of all, thank you so much. Absolutely incredible and very inspiring as another training as well. My question was, my original one was already taken, but I guess kind of building off of the question about commercialization and about, how do you actually expand cell therapies to where they are accessible and where this is actually feasible for people? You can't, obviously in the back of a CVS, you can't make a stem cell they're going to plant in someone's mind. I guess, how do you envision, what are the next steps in terms of making that feasible? Because you mentioned that was a big focus of, what makes BlueRock unique. And another follow-up question to that is, like you mentioned yourself, we've learned a lot from CAR T cells and, how successful those therapies have been and how far we've gotten in the clinical realm of implementing them. Do you imagine any sort of collaborations where, you might utilize the infrastructure that's being set up for making those treatments accessible? Is that something you could foresee as a collaboration, or do you have some other alternative approaches? How is this going to be feasible and to reach, planet Earth as you mentioned, Doctor?
Yeah, great question. Obviously that's on our mind a lot. I think the field and the community is not quite there, right? There's differences between countries. We have to work with regulators. That's like, it's a huge effort. You have to work in consortia, and it moves slowly. I guess this is the term I would use in a recorded webinar. But it's important, and we participate in those discussions because the regulators, both of the ones we work with, have been incredibly supportive of what we wanted to do. But they obviously also protect society from bad science. But we have to work with them to harmonize it. Harmonization between different countries will allow us to more easily bring the same therapy into other countries. Harmonization of companies that make shipping containers, delivery devices that are accessible to all companies will allow us to harmonize that, facilitating how we can bring this to multiple countries and more patients therein. So I think I think everybody's a little bit in their own world, moving this forward, what fits themselves. And everybody, I think, also is trying to move towards a common goal. But I think it is somewhat of a slow process that we're obviously trying to work on really hard.
Yes, hard and carefully. I really want to underscore that, the rigor and the need to not get another black eye. I mean, gene therapy lost years, Geron thing even with the cysts in the spine. I think that was a real black eye. Not to mention all the pirate MSC clinics where people are having to enucleate their eyes after getting treatment. So yeah, being careful, being measured, I think is a critical thing. And also, as we've said and described, this is a moonshot, right? So you made it to the moon, let's say. It's going to make everything move faster, I think, for everyone else. These intricacies, idiosyncrasies of the surgery that you're figuring out. I mean, this is the toughest. It's the brain. It's everything that I would imagine could never happen. 25 years ago, as I start out by saying, we were counting CFOS, C-Jun positive cells in brain slices in rats. And a quarter century later, these cells are going into people. I mean, I wouldn't ask for it to happen any faster than that. I think that the real key is that it's being done the right way. And again, hats off to the whole team for that. Very well done. Anything else? Any other questions?
Hi, thank you guys. Very simple question. How did you come to name this therapy? Because I do Dale.
I have nothing to do with it. So Stefan has to explain that one.
Yeah, so a fun fact, there's actually an organization, it's INN, and of course I don't remember what the acronym stands for, but it's an international naming convention. And there's certain rules, so as you can imagine, it ends in cell, which implies that the therapeutic active ingredient is a cell. And then there's certain rules about what other pieces of that word you can use. Then they suggest which letters you shouldn't use because they may not be available in all alphabets. And then you go through a process of coming up with suggestions. And then they, that you can really suggest many things, like you could put your name in there. So for us, the DA part was always we always referred to as DA1 or DA neuron. So that was the D. And we thought about balance, and then through various things that we couldn't say because it was already taken or it meant something else, we then ended up with the suggestion of bemdaneprocel. One of our clinicians liked that it rolls off easily when the physician prescribes. Sorry. One important difference, he would say, physicians will call it bemda.
I see.
Yeah. And then there's a process, and don't quote me on the exact, but there's a period of review where other companies and other people in the world can say, no, that's my name, I can't use that. And then finally you get a letter say, like, you can now call this bemdaneprocel.
Bemdaneprocel, what a beautiful series of syllables.
That's how it is.
I think actually Elon Musk named his 13th kid. Oh boy. Well, I guess that brings us to the end of the show. I'm sad. It's so much fun. I want to thank again our guests, Dr. Viviane Tabar, Dr. Stefan Irion. Also, thanks to BlueRock for hosting us. Amazing view. Never saw a seaplane in New York before. And STEMCELL Technologies for putting it all together. Thank you all so much. Also in the audience, it really made it special to have you here reacting and inspired, I think, like I was by these stories that were shared and all the details I think we want. So give yourselves a round of applause. You guys have been really good. Also, please be sure to check out our website at www.stemcellpodcast.com or your favorite podcast app to find this or any of our reams of great science conversations. You can also reach out to us via email at info@stemcellpodcast.com or via Twitter, now known as X. Elon. At Stem Cell Podcast is the handle. You won't find me on there, but Arun is very prolific. Thanks again, you guys, for listening, and good night.